Hello and welcome to this e-learning video. For this video, we're going to be talking about the new functions that were added into the version 23 Simcyp-R package. Specifically, we're going to be talking about how you can extract summary statistics and generate ribbon plots from your Simcyp simulations from R. So on YouTube currently, there are videos that have been created around the Simcyp-R package. I've also pasted a link in this video. I do highly recommend that you go to this video and watch them. These videos describe how you can use the Simcyp-R package for different scenarios, and it goes into more detail as to what the package is and what it's capable of doing. On members area, we also have e-learning videos that have been uploaded. So where can you find help? Within the package, there is a help page which consists of all the functions and their descriptions. It also comes along with example scripts and how you can run each function. We also have a vignette page which comes along with different case study scripts. So for example, there's a parameter estimation script that you can run, as well as a plotting script and also a sensitivity analysis script. In order to request for the package, please send an email to simcyp.support@certara.com, and they would add this package to your members area so you can download and use. Here are the list of functions within the Simcyp-R package, and what we can see highlighted in blue are the setup functions. So these are the functions that is needed in order to run a basic script using the Simcyp-R package. The next set of functions are the getters. So what these functions do is that they go into the Simcyp workspace and they extract parameter values for you. So for example, if you have forgotten the dose that is used within the workspace, you can use the function getDose in order to extract the dose value that is within that workspace. The next set of functions are the setters, and these functions, they set parameter values within the workspace. So for example, if you wish to change the dose value within the workspace that is attached in R, you use the function called setDose in order to change the dose value. The next list of functions are the output DBs. For these functions, they have at the end of the function name _DB. So what these functions do is after you run a Simcyp simulation, they are able to go into the database file that was generated and extract simulation results from that database file. So for example, if you wish to know what the simulated age is or what the simulated body weight is for your population, you use the function getIndividualValue _DB in order to extract that information from that database file. The next group of functions are the plotting function. These functions were added in version 23, and we can see that we've got three functions. The first is the plot forest DDI function, plot profile CI _DB function, as well as plot profile percentile _DB function. The use of these functions is to create plots of your Simcyp simulations. Finally, in version 23, we added summary statistics function. So for example, you can see that we have get compound results stats _DB, get forest data _DB, get individual value stats _DB, get PD results stats _DB, and finally get profile stats _DB. What all these functions do is that they give you the summary statistics of, for example, the profiles. So for example, if you're interested in knowing what the mean of your simulated profiles are, you use the function called getProfileStats _DB in order to extract the mean of your profile of interest. Now we're going to dive into our demo. For this demonstration, I'm going to show how to extract concentration time data for the full population. And then I would show how to plot individual profiles and then extract the systemic concentration profile of the mean, lower, and upper percentile. After this, I will show how to create a ribbon plot of the systemic concentration profile. And then I would show other summary statistical functions that are available within the Simcyp-R package that you can use. Okay, so now I'm going to go and open up my script. At the top here in this text window, I have already pre-written the script for this demonstration. So what the first line is doing is clearing my global environment. So if this was populated with information and different objects, running this line, line 3 would clear the global environment and make sure that it is empty. To run a script from R, all you need to do is click on the line that you're interested in running, and you can click on the run button, or a shortcut for running a line in R is Control Enter on your keypad. The next thing we're going to do is to load the necessary packages. So for the packages that I'm going to use for this demonstration is the Simcyp package, RSQLite package, and then for our plots, I'm going to use ggplot2. So now I'm going to run these 3 lines. That is now complete. The next thing that is important that I do is to initialize the Simcyp package. For this demonstration, I'm going to be initializing to the Simcyp Human Simulator. Next, I'm going to set my script to the source file location. For this, I'm going to use the function called script location in order to extract the location of where the script is saved. And once I've extracted that information, I'm going to save it into an object called path user. So as you can see on the right hand side, this is the path where I have this script saved. Once I have extracted the path of the script location, I am now able to set my working directory to that path. So next, I am now going to set my Simcyp workspace. So now what I'm going to do in my script is I'm going to use the function called set workspace in order to bring in that workspace into my R script. Once that is done, it would spit out a summary of that workspace for me. So it's telling me in this case that I have brought in the workspace default DDI version 23. And here is a summary of that particular workspace. So the substrate is SVL fentanyl, and it uses atom absorption model, where the distribution is minimal PPK, and so on and so forth. And the same for the inhibitor 1. The inhibitor compound is SV atazanavir, which also uses atom absorption model, and so on and so forth. The next thing I'm going to do is I'm going to now run a simulation. For this, like I said in the presentation, we use the function called simulate, and within this function we specify the database file name. So in this case, I can call the database file name whatever I wish. So for example, I can call this test1. So once I run this function, the simulation currently is in progress. I know this with this stop button showing on the screen. Once it is completed, the stop button will disappear, and then I'm able to run the next line. So now the simulation is complete. I am made aware that the simulation was successful. At the bottom here saying it is true. The next thing I'm going to do is to make connections to that database file that has been created. So previously I changed this to test1, so I need to make sure is when I'm creating my RSQLite connection, I need to make sure that I have the same name of the database file. So once I run this, the connection path has been made, and then now I'm able to go into that database file and extract simulation results. The first thing we're going to do is to extract the concentration time data for the full population. For this, the first thing I'm going to do is to extract the subject number. For this, I'm going to use the function called getParameter function, where the parameter tag for population size is simulation parameter ID $POB, and the category ID is $simulation data, and the compound ID is for substrate. So for this, we can see that I have 100 subjects within my workspace. Next, I'm going to create empty vectors to store my profiles for each subject. So the empty vectors I'm going to create is one for group, which will be representing each subject. Then I'll create an empty vector for my time profiles, empty vector for my systemic concentration profiles without inhibitor, so without the interaction of the inhibitor. And then finally, an empty vector for the systemic concentration profile with interaction. So now you can see on the right hand side, I've created empty objects, which will then be populated later on in my for loop. So in this for loop, I'm collecting the time profile for each subject, and then I'm collecting the systemic concentration profile. We use the function called getProfileDB. For this function, we specify the profile ID. In this case, we're interested in CSIS. Then the individual number. In this case, this will be iterated based on the for loop. So the first subject will be subject 1, so I is equal to 1 in this case. Next, we need to specify the compound ID. In this case, I'm interested in the substrate concentration profile. We mentioned the RSQLite connection, and finally, if we wish to see the profile with or without interaction. So the first case is the profile without interaction, hence why inhibition is equal to false. And the second case is for me to extract the concentration profile with interaction, hence why inhibition is equal to true. So once I've extracted these two profiles, I'm saving them into an object called currencies, and then currencies inhib representing the systemic concentration profile with interaction. After that is done, I'm then saving my data into the object that was empty previously. So I'm saving the group numbers, in other words, the subject's number, the time for each subject, and the systemic concentration profile without interaction, as well as the systemic concentration profile with interaction for each subject. So now I'm going to run my for loop. So now that my for loop is done, I can see on the right hand side a once empty object called CSIS is now populated with a large number of data. The next thing I'm going to do is to clean up this data. So I'm going to combine all the data and save them as a data frame. Once that is done, I now have a data frame which looks like this called Simcyp-R, where the first column represents the individual number. The next column is a time for each subject. The next column is the systemic concentration profile without interaction for each subject. And finally, the last column is the systemic concentration profile with interaction for each subject. So now I'm going to make a plot of the individual profiles. To do this, I'm going to use ggplot. So what we can see on the right hand side is I've created a plot of my individual profiles, both for the systemic concentration with and without interaction. The next thing I'm going to do is to extract the mean, lower and upper percentile of the simulated systemic concentration profile. So for this, the function I'm going to use is getProfileStats_db. For this particular function, we need to specify the profile ID that we're interested in. So in this case, we're interested in the CSIS. And then we need to specify the compound ID, in this case, the substrate profile. For inhibition, we're going to set it, first of all, to equal to false, meaning that we're interested in the profile without interaction. And then we are also going to mention which percentile level we're interested in. So in this case, for the upper percentile, we're looking at the 95th percentile, and we want the 5th percentile. And then also we need to specify whether we want the trial statistics or not. So for this example, when the trial is equal to false, this means that we are returning the population statistics. And then finally, we need to specify the RSQLite connection. So once I run this line, it's telling me that it's returning the population mean profile. The first code in line 88 is the population mean profile without interaction, whereas in line 91, it's the mean population mean profile with interaction. So in this case, we have inhibition set to true. You can also choose to view the results on the trial level. So instead of viewing the summary statistics for the full population, you can view the summary statistics of each trial. So what you would need to change in the function is you would need to set trial is equal to true. So if I run this line, it will tell me that it is returning the trial mean profiles. Once this is complete, I would see the results on the right hand side. So clicking on this, I can now see I have the mean profile for trial 1 as well as the lower and the upper percentile for trial 1 and for the other trials within the simulation. The next thing I'm going to demonstrate is how to create a ribbon plot. To create a ribbon plot using a Simcyp-R package, we use the function called plot profile percentile underscore db, where we need to specify the profile ID. In this case, we're still interested in the systemic concentration profile, as well as the compound ID, whether you want to view this for interaction or without interaction. So the first one is for the profile without interaction, and the next code is for the profile with interaction. And then you also need to specify the upper and the lower percentile level that you're interested in. And then finally, the RSQLite connection. You can also choose to color your ribbon plot. So running this line, we can see that this is our profile without interaction. To view the profile with interaction, all we need to do is set the inhibition to equal to true. Once a profile is generated, we can see the profile in blue is the systemic concentration profile with interaction, and the profile in red is the systemic concentration profile without interaction. You can also choose to plot the profiles without the ribbon plots. So in this case, we're just going to see the mean profile. To do this within our function, all we need to do is add a new argument called show ribbon and set this to equal false. So now we can see and view our profile without the ribbon plot, without the percentiles. The next thing we're going to do is talk about other summary statistics function available within the Simcyp-R package. The first is if you were interested in demographic statistics. So for example, if you were interested in knowing what the mean age that was simulated is, or the mean body weight. For this, we use a function called get individual value stats underscore db. To run an example, simply go into packages and search for the package called Simcyp. Once you're within the help file, you can then search for the function get individual value stats underscore db. And as mentioned in the presentation, you get the description of how to use the function, as well as the usage, as well as description of each argument. And finally, if you scroll all the way to the bottom, you will see example scripts. So for example, we have one for the age. So you can copy the example code and paste it in your script. I'm going to save this into something called stats age. Once I run this function, this will give me the summary statistics of the simulated age. So in this case, it is returning the trial statistics, and that is because I've set trials is equal to true. So if I was to view this data frame, I would see that it has returned the summary statistics for each trial. To view the summary statistics for the full population instead, within my code, I can change the trial to equal false. So once I run this function, it will tell me that it's returning the population statistics. And now I can go back into the data frame, and it is now telling me that this is the summary statistics for the full population instead. For compound statistics data, we use the function called get compound results stats db. So if I go back to the help page, I can search for that function. Clicking on that function, I get the description, similar as before, and then if I scroll all the way down to the bottom, there are example scripts, which I can then copy over. So the first example is for adjusted FU. So I could save this into something called stats FU, and then run the function. And then once that is complete, I can view the data frame of the summary statistics. Assuming I had PD activated within my workspace, this will generate PD results for me. To view the PD statistics, we use a function called get PD results stats underscore DB. An example code is also found in the help file, where if I navigate to that function and I scroll all the way down, these are examples of how you can run this function. Here comes the end to this e-learning video. Once you're done with your R script, it is important that you make disconnections to the database file as well as on initializing the Simcyp engine. Thank you very much for your time.